Okay, so welcome to the video on chapter 4, where we're going to discuss the basics of kind of the scientific method. So we'll go over all the processes and kind of parts of the scientific method, what it is, and why we complete it. And then we'll go through a general kind of chemistry discussion where we're going to talk about matter, we're going to talk about energy, and we're going to look at kind of how energy is converted from one form to the other. And then all this is important basically because in order to understand a lot of the concepts that we're going to talk about in later chapters, so for example, photosynthesis, respiration, which we will mention today, but by understanding how kind of that's interrelated with environmental systems and how energy is converted, say, from um, sunlight into carbon molecules, which is then used to fuel our muscles. You have to understand how energy is converted to one form or the other. Right, so we're just going to kind of use this chapter as kind of a basic foundation for some of these scientific principles. Okay, so we're going to first go over the nature of science. We're going to talk about some limitations that it has, pseudoscience, the structure of matter, and we're going to look at the principles on energy, and then we'll look at kind of energy flow in the environment and some implications of that. So there is or are these cause and effect relationships, right? So one common example of a cause and effect relationship is wood stoves cause air pollution, right? And one of the way that wood stoves causes air pollution is from the burning of the wood that you put into the fireplace, and it releases smoke that has different sizes of particles that go into the atmosphere, and then they tend to circulate in the atmosphere. Now, it might not seem like much for one person burning wood, but whenever you have, millions of people around the world burning wood, it tends to add up and tends to have a big effect on particulate matter in the atmosphere. Now, because of this, the EPA set standards for wood stoves in 1990. But one of the big downsides to this standard is that 3/4 of the wood stoves in the United States, for example, were installed before 1990, and those wood stoves can and are still being used today in some of the older houses that they were installed in. And so this is just some examples of kind of community and state laws that kind of have an effect on this cause and effect relationship. And now we can generate cause and effect relationships through the scientific method. Okay? We can figure out kind of what is leading to an observation that we've seen or made in the natural system or the natural world. And in kind of a broad general sense, the basic foundation of science is this process that's used to solve problems or develop an understanding of nature that involves testing possible answers. So basically, in a nutshell, in science, you make an observation, you're curious about why that observation happens, and then you go and test possible answers to that observation. But with science, we want to do this in a more rigorous format, not kind of this random testing, you know, that doesn't have a lot of validity to the results. So, what science does, or what science has done, is we've developed this method, the scientific method, where we can follow this rigorous testing format to determine if our proposed solutions to some observation that we've observed is actually valid, if there's evidence supporting the validity of that observation. And the scientific method has this specific kind of outline and process that we need to follow in order to make sure that we're following kind of the most rigorous standards that we can in the community. And the basic outline for the scientific method is you have these specific events that you've observed. And by observing these events, you can kind of get at some of the causes that may be identified. So, for example, let's say you have two shrubs in your yard or a garden, right? One shrub may have purple flowers, another shrub may have white flowers. And you notice as you kind of spend time in your yard that you only see bees going to the purple flowers, and you never really see bees landing on the white flowers. And so you've made an observation, and you can be like, all right, well, that's kind of weird. How come they only go to the purple flowers and not the white flowers on the shrubs? So now you can generate some causes, and you can maybe say, all right, maybe they see a specific wavelength in the color spectrum. Maybe the flowers produce different types of nectar or different scents. And so you can start to kind of guess at some of these causes, and then through the scientific method, you can identify some of those potential causes. Then, once those causes have been identified, you can generate rules or patterns to describe these observations. And so let's say, for example, that the bees only can see in the visible wavelength of, say, the purple color, then in general, you would expect bees to only visit flowers that fall in that range of wavelengths that they can see. And so you would repeat these observations, repeat that experiment to see if your general rule or pattern holds true across different bee populations. And so you want to make sure that these perceptions that you make and create are individualistic, right? That there are, they're made from kind of this body of work that you've put together, and not just really this one specific study that you use to say, all right, this is how it is, and these are the causes. And what's cool with this is that we can make fundamental rules of nature that are universal. And so all of these assumptions here are kind of grounded in science and the scientific method. And so we can go in expecting these assumptions to hold in the process or through the process of science. Now, in the first slide, I mentioned kind of this cause and effect relationship. So the wood burning stoves causing pollution. Now, we can distinguish between situations that are correlated, meaning they happen together, and those that are correlated and actually show a cause and effect relationship. And so it's important to understand these relationships, right? So just because something's correlated does not mean that it causes that thing to happen. So the big thing you want to remember is that correlation does not mean causation. Right? Let me give you an example. This is one of my favorite examples of this. So let's say there was a study, and it says that if you eat more ice cream, you have a higher chance to be eaten by a shark. And those chances are correlated. So basically, if there's very little ice cream eaten, then that means that you have a lower chance to be attacked by a shark. If there's a lot of ice cream eaten, then you have a higher chance to be attacked by a shark. And you're probably thinking, well, how did they get that to be correlated? And it was actually a pretty strong correlation. And you wouldn't expect the amount of ice cream you eat to be the cause of shark attacks, right? and so they're not. So this is the whole point of this kind of relationship, and the researchers bringing this up. In fact, it's just a coincidental correlation between the amount of ice cream eaten and the risk of shark attacks, because if you think about it, more people go to the beach during summertime. Summertime, it's hot. What do you do when it's hot? You eat ice cream. So if there's more people at the beach during summertime and they're eating a lot of ice cream, that means there's more people in the water. And so there's more of a chance of a shark attack happening because there is more people in the water, right? And so just because a lot of ice cream's eaten doesn't necessarily say that you're going to have a chance of a shark attack. Okay, so just remember, correlation does not equal causation. So the cause of the shark attacks was completely not related to eating the amount of ice cream you ate. Okay, and there are lots of examples of this in the scientific world, even not in science, just in statistics in general. You can find correlations that are not where one variable is not causing the other, they're just related. Now what we're going to do is just kind of go through the steps of the scientific method. So like I mentioned, the scientific method requires this systematic setup. And this is where you're looking for information on an observation or a question that you've proposed, and you're trying to figure out the cause of that observation or mechanism or behavior or whatever you've observed. And so an interesting thing with the scientific method is that it's always undergoing reevaluation, criticism, and modification. Um, there are a lot of ideas in science that are around for a very long time. All of a sudden, new evidence is brought to light, and then we change that idea. One example is when I was in high school, we learned about the five kingdoms, so the hierarchy of organisms, which we'll learn about in the next chapter, I believe it is. But basically, your kingdoms used to be the largest kind of group of animals. So you had like the animal kingdom, the plant kingdom, the fungi kingdom. But now, new research has shown over the last decade or so that there's actually a bigger classification above kingdoms that are domains. So you have like a domain for bacteria, a domain for archaea, and then a domain for eukaryotes. And then that domain of eukaryotes holds the five kingdoms that I learned about. So that's kind of an idea um, uh, that has been modified in my lifetime because of new evidence that's been brought about through the scientific method. So the general kind of flow of the scientific method is that you make an observation, you then ask a question, right? So let's go back to our bee example. We noticed that the bees were only going to purple flowers. We're like, well, why are they only going to purple flowers? What do they not like about the white flowers? We form a hypothesis. My hypothesis is that they only can see colors in the purple visible spectrums, right? So that wavelength of light. So then what I could do is test that hypothesis. There are ways where you could measure and test kind of how bees respond to different wavelengths of light, and you would get that results or those results, and then you would make your conclusions, draw your conclusions. And if the data fit with your hypothesis and current scientific theories and laws, you would show that it supports your observation and supports other theory. If not, then you can say, okay, well, if it's not the wavelengths, then maybe it's the nectar that is in the flowers. Then you could go in and test if that nectar is different, and if the bees get different, different outcomes by collecting and bringing that nectar back to their colony or hive or whatever. And then you can look at those results. And so you're constantly drawing conclusions, revising your hypothesis. And so you're going through this kind of cycle, right? Where you're testing, making conclusions, redrawing your hypotheses. And a lot of the times when you get your results, even if you have evidence supporting your results, supporting your hypothesis, it may bring up new questions. You may see something interesting in your results and you may be like, man, I wonder why this is the case. Let me look into this even further. And so then you have a new question, and then you kind of make a hypothesis about that. And so you kind of keep this iteration going. And at the same time, you're going through all of these steps. You're also communicating with other scientists. You're going to conferences. You're, sharing your work. You're talking with other scientists about the theory. You're seeing what they think about your observations. And that's allowing you to get input on ideas and methods that you probably wouldn't have thought of or wouldn't have considered if you wouldn't have collaborated with others. And so that's why it's really good to kind of involve and network in the scientific field that you're working in. And so just to kind of put all of this down in text for you, I know we went through a lot of it in the figure here, so we'll kind of run through this pretty quickly. So first you start out with an observation, right? That's just an event you record with your senses, right? So you just observe that event. And we generally refer to those observations as careful, thoughtful recognitions of events, right? My bee example would be a thoughtful recognition of, how bees interact with flowers. Observations usually lead to multiple questions. And so you can ask one question and answer that one, and look at the knowledge and information that you get so that you can kind of answer the other questions and continue to gain information. Now, from those questions that you get from your observations, you need to make a hypothesis. And it's very important for your hypothesis to be testable and to be able to be proven false. So the big thing with a hypothesis is that it has to be able to be falsified. If you can't prove your hypothesis to be false, you need to reword it or rework it, because the data that we're collecting is for that reason, right? To prove your hypothesis false. If you can't prove your hypothesis false, then that suggests that the data supports your hypothesis. And so what you want to do is you want to keep gathering evidence through more scientific experiments. And as long as you don't prove your hypothesis false, we assume it to be true, because we never really can prove something to be true. So the way we assume that it's fact is by gathering all of this evidence that shows that we haven't been able to falsify it. And so, again, your hypothesis needs to be logical, needs to account for current theory, current scientific information, and it needs to be testable. Okay. After you generate your hypothesis, then you develop an experiment. You can do controlled experiments, which basically divides your experiment into two groups. One group will be a control group, the other will be an experimental group, and they'll really only differ by one variable. And like a really easy example of a controlled experiment is, let's say you've manufactured a drug that is supposed to help regulate high blood pressure. In order to see if your drug works, you would get a population of people that are from varying cultures, ethnicities, genders, races, and working classes. And so you want to get, basically all the variation that you can get in human beings so that you account for that, all of that variation. Okay. And so once you get your group, you'll then randomly divide it into two groups. One will be your experimental group, one will be your control group. So you'll divide that sample in two. Your experimental group will get the variable changing kind of experimental method, and that would be the medicine itself. So the experimental group would actually get the medicine that is supposed to lower the blood pressure. The control group will not get a medicine that controls for blood pressure. They would get a placebo. And the whole goal is to not let the participants know whether or not they're getting the experimental drug or the control, because you don't want them to change anything about their lifestyles, right? You want them to act naturally. And so the variable that we're changing is the drug that they're getting. They're either getting it, which is the experimental, or they're not getting it, which is the control. So then you would check their blood pressure. And so if the average blood pressure of your experimental group is lower than your control group, then you would assume that your drug lowers blood pressure. And so that's the difference between an experimental and control group. Now, you can do observational experiments, so you don't necessarily have to do a controlled experiment. It just depends on the type of question and the type of experiment that you want to conduct, and the question you want to answer. The big thing, though, whether you're doing controlled observational experiments, is that they're able to be reproduced. That you can say, if you're observing a group of animals, that you can go and observe those animals over and over again. Somebody else could go and observe those animals, do the same thing you did, and reproduce your experiment. Same thing with the controlled experiment. Another scientist could go and get a different group of people, do the same thing you did, and replicate that experiment to see if they get similar results to you. And that's really important, because if you get one result and somebody else gets the same result, it supports your ideas even more. If they get different results, then it brings up the question as to why, and it may bring to light some new observation or relationship that you might not have caught or seen in your first study, and so you can go back and look at it. Now, the next part of the scientific method, or basically science in general, that we're going to talk about is theory versus hypothesis and a scientific law. One of the big issues with the public and their view on science is that a lot of the times you'll hear somebody say, "Oh, it's just a theory, so that doesn't mean that it's, that big of a deal." Or they'll say, it's just a hypothesis. Okay. The latter one, it's just a hypothesis, is the more correct statement, because a theory is one of the strongest kind of foundational ideas you can get in science, because a theory is pretty much widely accepted in that scientific field. It provides a generalization about some fundamental scientific concept that explains why something happens, or why an observation happens, or some other process in the natural world. And theories have years and years of evidence that's backing up that idea. A hypothesis is only made from one observation, and it is a educated, rationalized, kind of I just lost my train of thought. It's a rationalized way to go about explaining your observation. Okay. And so it hasn't had a lot of evidence, a lot of support, decades of work. And so it's really not strongly supported yet. You can do science on your hypothesis, and others can expand on your hypothesis. And eventually, if there's a lot of support, it can become a theory. So a theory is one of the strongest ideas we have in science, and so is a scientific law. So a scientific law is this uniform or constant fact of nature that, again, like a theory, is describing what happens in nature, whereas a theory is explaining why things happen, right? And so, the next time you hear somebody say, oh, well, it's just a theory, you can say, well, that's the strongest ideas we have in science. So that's just something that, is a little pet peeve of mine, that people kind of throw that word around and don't really understand exactly what it means and kind of what it stands for. And so that's where communication really comes in terms of, the scientific method and how we get this information to the public. Um, so public understanding is a big issue, like I just showed with the whole theory versus hypothesis and scientific law. But it's also important to get our research out to the public world, and we do this through the publication of articles in scientific journals. And these publications are peer reviewed. Other kind of experts in our fields will review our papers. They criticize our papers. They make suggestions, or they agree with our work and agree that it should be published and put out to for the public to read, and basically the rest of the scientific community. So this peer review process is one of the most rigorous forms of publication for scientific work. Whereas, things like news articles, or, some magazines, they can contort and kind of distort the message that a scientist may be trying to give, and kind of word it in a way that's not correct. And so, observations, scientific query begins with it, an event that's occurred, and then we basically end with communication, where we're publishing our research, publishing our ideas, our understanding of events and observations, so that the rest of the scientific community and the public community um, can kind of read and hopefully understand and take this new knowledge and information. Now, even though science has this kind of rigorous setup in the scientific method, there are some limitations to science. Every scientist struggles with the same moral and ethical questions as regular citizens. One of the big things is that, as people who are reading this work and interpreting this work that's published by scientists, is that we need to be able to really differentiate between data collected during, say, an investigation or an observation, and the opinions of what this data mean. So a scientist can collect results, but they will give you an interpretation of those results. That doesn't necessarily mean that that's a fact of how those results should be interpreted. Right? So they can be interpreted incorrectly. And so that's where you kind of need to understand what is exactly the data versus what are the opinions and the conclusions and discussions about the results that come from the scientists themselves. And so you want to be able to recognize that scientific knowledge sometimes gets used by different people to support opinions that may not be valid. So again, they can kind of contort what that data is actually saying to support their opinions in an incorrect way. And this kind of falls under the idea of pseudoscience, which is basically a deceptive practice that uses the appearance or language of science to convince, confuse, or mislead people into thinking that something has scientific validity when it doesn't. So basically that they're taking some results, kind of wording it, misinterpreting it, um, um, and making the public believe that it's saying something that it really doesn't. Okay, so that's pseudoscience. And so that's kind of the basics of science and the scientific method and kind of how we generate our ideas and experiments and then get it to the outside world that's not part of the scientific community. What we're going to do with the rest of this chapter is look at the structure of matter, and then we're going to talk about energy, and then we're going to talk about some implications of that energy and in the environment. So in general, matter is anything that has mass and takes up space. Okay, so our smallest types of matter are atoms. Okay, so all matter is made up of at least one or more um, atoms, and these atoms are always in constant motion. And atoms have a nucleus and they have an electron shell or electron cloud. Okay. The nucleus has protons and electrons. Protons have a positive charge and they have a mass of 1, and neutrons have a neutral charge. They also have a mass of 1, and electrons have a negative charge and they have no discernible mass. So they basically weigh nothing. So the atom, right, is that fundamental unit of matter because, like we said, matter is at least one atom or more, depending on what type of matter you're looking at. There are 92 atoms found in nature, so naturally occurring atoms, and each atom forms a known element. So, for example, like carbon, hydrogen, helium, boron, chlorine, sodium, potassium. So all of those are an element that are formed by a specific type of atom. And like I mentioned, atoms are protons, neutrons in the nucleus, and then electrons in that electron cloud. Now, all atoms of the same element have the same number of protons and electrons, but they can vary in the number of neutrons. Okay. Now, with with atoms, you can determine their atomic mass by how many protons and neutrons they have. Okay, so if you looked at a periodic table, right, you would see a bunch of rectangles with an element symbol, with the atomic number, and then the atomic mass. Let me fix this number up here. Okay, this atomic number is 6. All right, so carbon has an atomic number of 6 and atomic mass of 12. So basically what this is telling us is that carbon has 6 protons, 6 neutrons, and 6 electrons. Okay, now remember we said that electrons don't weigh anything, so they don't add to the atomic mass. We said neutrons have an atomic mass of 1, and so do protons. So if you have 6 protons and 6 neutrons, then you have an atomic mass of 12. Okay, 6 plus 6 is 12. What's interesting, though, is that you can get isotopes of atoms that differ from one another in the number of neutrons that they contain. So, for example, the most common carbon atom is carbon 12, So a carbon that has an atomic mass of 12, so 6 protons and 6 neutrons. Now there are rare forms of carbon atoms that are known as carbon-13 and carbon-14. You may have heard of carbon-14 before because carbon-14 is used to radiocarbon date fossils and sediments. And the difference in carbon-13 and 14 is their atomic mass, 1. So carbon-13 has an atomic mass of 13. Carbon-14 has an atomic mass of 14. The way these atomic masses differ is that they differ in the number of neutrons in the nucleus. So carbon-13 has, I'm going to write it over here. So 13 has 6 protons still. Protons never change, but it has 7 neutrons. Carbon-14 still has 6 protons, but it has 8 neutrons, right? So 6 plus 7 is 13, 6 plus 8 is 14. And so that's what isotopes are. Isotopes are just variations of an atom of the same element that differ in the number of neutrons, and so they have a different atomic mass. And one interesting thing, this is a side note, you won't be tested on this, but these atoms that have more neutrons are heavier, so they actually move slower and accumulate slower into, say, animal tissues versus carbon-12. And so you can actually look at the number of carbon-14 atoms in an animal, and you can actually tell, like, did they favor certain types of grasses or plants? Because plants assimilate carbon differently. And so you can use kind of the weight of those carbons to figure out what an animal ate through like a fossil tooth or a fossil bone, which is pretty interesting. So that's how isotopes are really useful. Clear these off. Okay. So atoms can be bound together to make molecules. Okay. So ions basically are electrically charged particles. So you can have an atom that is positively charged, or you can have an atom that is negatively charged. Okay? And this follows the basic rules of opposites attract. So you'll have a positive atom attract a negative atom. So for example, let's see what we have here. Sodium is positive, and chloride is negative, right? So if you have sodium that's positive, chloride that's negative, they will bind together to form a molecule that is salt, right? That is sodium chloride, okay? And so those ions, those electrically charged particles, will attract one another. And when you get 2 or more atoms or ions to bind to one another, you get compounds. Okay? And so again, example, sodium chloride or water or table sugar, mixtures of variable combinations of atoms or ions. So for example, honey, concrete, air, all of those are different examples of mixtures. Okay, so compounds, again, atoms that bind together, 2 or more bind to one another. Mixtures are combinations of atoms, ions, or molecules, and that varies depending on the type of mixture. Next, just want to kind of mention water and how important water is to life. We know that water can exist in 3 phases: solid, liquid, or gas. We know Earth, 75% water covers the surface. And water really determines kind of the weather and the climate in a particular area. And the flow of the water, flow of ice, flow of glaciers really shapes the Earth's surface. So, for example, if you have been to the Bay Area, lived along coastal California, you know that it's a lot cooler during the summer on the coast than it is in the interior. That's because of the ocean. In the wintertime, or I should say, yeah, in the wintertime, the water's cooler than the land, and so it takes up all of that heat and mitigates it from the coastal areas. In the in the wintertime, it's the opposite. The water has all of that heat from the summer and it releases it during the winter, and so it keeps the coastal areas about 70 degrees, right? And so the average temperature for say San Francisco, doesn't change that much from summer to winter, whereas say the average temperature for Merced, goes from like 50 or 60 degrees in the winter to 90 degrees, during the summer for average. And so water is really big for regulating climate in a region. And it's also the most common molecule found in living things. And it plays a really big part in how objects or how ions and molecules can move throughout our bodies. Water is an interesting molecule in that it has it's a polar molecule. What that basically means is that water has a positive end and a negative end. So let's say we have a water molecule here, H2O. So one oxygen atom with two hydrogen atoms bound to it. Now, oxygen is a greedy electron acceptor. It likes to hold the electrons around its nucleus more than the electrons spin around the hydrogen nucleus. So what that does is, because those electrons are negative and they spend more time around oxygen, the oxygen end of the molecule is partially negatively charged, whereas the hydrogen atoms have a slight partial charge. Okay, so what you can imagine is another ion that comes into contact with it. So let's say like sodium chloride, Na plus, Cl minus, right? Salt gets put in water, it dissolves because of water's polarity. So the hydrogens will grab the chlorides, and then the oxygen will grab the sodium and bind to it. And so it pulls those sodium and chlorine crystals out of that salt molecule and basically dissolves it. What's also cool is oxygen, other water molecules will orient themselves to where, say, the oxygen has what's called a hydrogen bond with the hydrogen molecules. And so they'll kind of orient themselves this way, right? Then if you had another water molecule here, have like a little bond. Here's the other hydrogens. Right? And so you can get this network of water molecules. So this is why water is liquid at room temperature, because of the way these water molecules interact. And you need energy to break these hydrogen bonds. And once those hydrogen bonds break, the water molecules will separate, right, and go from a liquid water to a vapor. And so that's why the evaporation of water off of our skin can cool our bodies, because the heat from our body is going into breaking that hydrogen bond. And then as that bond breaks, it's lost, and it's lost from our body. And then the water evaporates as sweat. And so again, like I mentioned with the salt, water is what's known as a universal solvent. So it can dissolve a lot of different molecules and substances within it, which makes it very important. So here's some relationships between the different subunits in matter. So for example, an atom has those subatomic particles we mentioned, protons, neutrons, electrons. Elements are made up of atoms. Compounds are made up of molecules or ions. And then mixtures are made up of atoms, molecules, or ions. Okay, so it's important to know the distinction between these different categories of matter. All right, so what we're going to talk about next are acids, bases, and the pH scale. So in, say, a solution of water, you can have hydrogen ions. Okay, so hydrogen ion would just be a hydrogen atom, right? And they generally have, positive charge. Now, if you have a substance that when it is put into water, it releases this hydrogen ion, then it's known as an acid. If you have a substance that takes up hydrogen ions from that solution, then it's known as a base. We can measure the concentration of an acid or a base, basically how much or how high the hydrogen concentration is in a solution through pH. And so that's the measurement. So the pH is basically the measurement of the hydrogen ion concentration in a solution. The pH scale, which is what we would measure that solution on, ranges from 0 to 14. If it's 0 to 6, then it's acidic. It has a lot of hydrogen ions. If it's basic, it would range from 8 to 14, and it has fewer hydrogen ions and more hydroxide ions. Or hydroxide molecules, right? Because you have an OH, right? So acidic, fewer OH, more hydrogen. Basic, more OH, less hydrogen, okay? So the hydrogen concentration determines whether it's acidic or basic, or also alkaline. Base is alkaline as well. So here we have a pH scale. Again, going from 0 to 14. 7 is neutral, so that's pure water. So hydrogen ions and hydroxide ions are equal. You go below 7 on the pH scale, you get more acidic, more hydrogen ions. Examples are, coffee has a pH of 5, tomatoes in between 4 and 5, soft drinks, pretty acidic, around 3, 3.5. And then gastric juices are, lower than 3 to about a pH of 1. So very, very acidic. Now, basic solutions are things like your cleaners, so like bleach, ammonia, usually things that you take to help with like acid indigestion, are the chalky taste is because you're getting a basic solution or compound to take up those hydrogen atoms and to remove them from your digestive system. Now, one thing I forgot to mention on this slide is that the pH scale is inverse and logarithmic, okay? Basically what this means is that as you go down in the pH scale, the concentration of hydrogen ions goes up, right? As you go up on the pH scale, the concentration of hydrogen ions go down. Okay, so that's the inverse. The logarithmic comes in that every change in every 1 unit of change on the pH is a 10-fold change in hydrogen ion concentration. Okay, so let's say you have 10 hydrogen ions in the solution at a pH of 6. Okay, so 10 at a pH of 6. What would be the concentration of the hydrogen ions at a pH of, let's make that 5? Okay, so what would that concentration be at a pH of 5? And so take a minute, pause the video, think about it. All right, hopefully you got 100, right? Because we've moved 1 unit on the pH scale. And remember, we are increasing in hydrogen concentration because we are going down in pH. So that would increase by 10-fold because it's 1 unit. So it would be from 10 to 100. Okay. Let's say we went from 6 to 4, right? We're making another step, so it's 2 total steps, right? So we would be at 1,000. Okay, so every step, remember, is 10 times. So if you do 2 steps, you're basically doing 100 times. You're changing by 100. Right? 10 times 10 is 100 times. Okay. So if we change 3 units, so if we went from 6 to 3, it would change by 1,000 times. So then it would be 10,000 hydrogen ions. Now, if we went from 6 to 7, right, you would get a 10-fold decrease. So we would be at 1. We went to 8, right? You would get another 10-fold decrease. So we would be at 0.1. So on and so forth, because you get less hydrogen ions when you increase in pH. Okay? So you want to remember that. pH scale is inverse and it's logarithmic. You know what I mean? It's a log scale. There we go. I can't talk. Looking at inorganic and organic matter, we refer to these and make the distinction based off of what that material is made out of in terms of their atoms. Organic matter is molecules that contain carbon skeletons. So they have these carbon atoms that are bonded to kind of form that skeletal ring or chain. So organic molecules can be rings. So like steroids form rings. Glucose forms rings in water, or chains. So chains would be like starches, which are long chains of glucose molecules in plants, and glycogen is long chains of glucose molecules in animals. And all living things contain these organic compounds. There's 4 major organic compounds that living organisms can have. Humans, or I should say humans, have these 4 organic compounds, and that's carbohydrates, lipids or fats, nucleic acids, and proteins. So those are the big 4 categories of organic matter. And chemical bonds in these organic molecules actually contain energy that can be released when the bonds are broken. So for example, when a glucose molecule is broken apart, the energy in those carbon bonds are released and then stored as ATP molecules, which can then be used for work within the body. And so chemical bonds are attractive forces between atoms that make an interaction between their electrons. When the chemical bonds are formed or broken, you get a specific reaction to occur. In exothermic reactions, bonds in the new compounds contain less chemical energy than the previous compound, so they've released energy. In endothermic reactions, the newly formed chemical bonds contain more energy than the previous compound, so they've stored energy in those products. Okay, so exo is releasing, endo is basically storing. So exo has less energy than the previous, endo has more than the previous. Now, when these reactions take place, there is a specific amount of energy that's required to be input into the system in order to make that reaction happen. This is known as an activation energy. And enzymes, which are proteins that are really important because they help to lower the amount of energy that's needed to start that reaction. So they essentially lower the activation energy necessary for a chemical reaction. And so again, a catalyst alters that rate of reaction, and it can be reused over and over again. And so enzymes are really important catalysts in our bodies. So here you have some different chemical reactions. You have methane and oxygen to produce carbon dioxide, water, heat, and light. And so this is the same reaction, just shown in different forms, basically. So first line is just words. Second line is the chemical formula, and then the third line is the stick figure formula, basically. Chemical reactions happen in living organisms, and like I mentioned, the enzymes are the catalysts that reduce that activation energy. And photosynthesis is one of the big chemical reactions that happen in plants. And basically, plants take inorganic energy through photon packets, so sunlight. And so they take energy in the sunlight and carbon dioxide and convert that into an organic compound, which is glucose. And then that glucose can be stored in plants as starch, which they will then use for their energetic needs in their cellular processes. And the formula for photosynthesis is C or 6CO2 plus 6H2O gives you C6H12O6 plus 6O2. So basically what this formula is saying is that you need 6 molecules of carbon dioxide, 6 molecules of water to give you 1 molecule of glucose and 6 molecules of oxygen. Okay? Now, whenever we take that glucose molecule in our bodies, say we eat a plant, we get that glucose molecule as a starch. Our body will break down that starch into individual molecules, and then we'll break down that glucose molecule through the process of cellular respiration. That, in combination with oxygen, will take that glucose molecule and make ATP, and that ATP is used for our cellular processes like moving products and ions and atoms in and out of our cells, making our muscles contract, all sorts of other processes. And so if you notice, respiration is actually the reverse reaction of photosynthesis. So you start out with glucose, you have 6 molecules of oxygen, that gives you 6 molecules of carbon dioxide, 6 molecules of water, and energy, which in this case is ATP, adenosine triphosphate. So here's just a figure of photosynthesis and that formula. And for cellular respiration, all organisms, so even plants, have to do some form of respiration, since they need kind of that energy. They need that energy to maintain their cellular processes. All right, so one of the last things we're going to talk about in this chapter is or are energy principles. So energy, in the basic sense, is just the ability to perform work. So work is done when an object is moved over a distance. So energy can be found in two different forms. You have energy that is in moving objects, which is kinetic energy, and then you have potential energy, which is the energy in an object based on its position. Okay, so here we have a picture of a dam. Notice that the water in this dam is higher than the water below the dam. So the water is up higher, and so it has a high potential energy because it wants to move downwards, right? So as they control the dam, they open the floodgates on the dam, they allow that water to flow through, and so as it flows down with gravity, the energy of that water moving, that kinetic energy, is being transferred to turbines. So that water is spinning a turbine, and the spinning of that turbine is making the energy. So the movement of the water, that kinetic energy, is being transferred into electrical energy. So you have the potential energy in the water being stored up high, and then when that water is allowed to move down with gravity, the kinetic energy is transferred in those moving objects. Now, the state of matter depends on the energy that's present in the object. The amount of kinetic energy contained in a molecule determines how rapidly that molecule moves. So solids have particles with low energy, vibrate relatively close together, don't really move kind of long distances. Liquids have more energy, molecules move farther apart from one another, and gases move very rapidly, very fast. Okay, so they're kind of bouncing all over the place, the molecules are. So in this image, we have a figure kind of showing that. So your solid, right, your molecules are tightly packed together, not really moving that far. In the liquid, there's a little bit of movement, a little bit of space, but nowhere near as much as the gas form. And in liquids, not as tightly packed together as the solid form. Now we can have what's known as the sensible heat transfer, which is when heat energy flows from a warmer object to a cooler object. So temperature of cooler matter increases and the temperature of the warmer matter decreases. Okay, so like ice, you put ice in your drink, the ice will increase in temperature because it's transferring, or the heat of the drink is being transferred to the ice, right? And then the drink will cool down. Okay, latent heat transfer is when heat energy is used to change the state of matter, but the temperature of the matter does not change. Okay. We also have some laws of thermodynamics, which is related to energy. So basically, the overall law or statement of energy is that energy can be converted from one form to another, but it cannot be created or destroyed. So the amount of energy in the system remains constant. It's just being converted to different forms. And so that first law of energy states that energy can't be created or destroyed, only can be changed from one form to the other. The second law says it can be converted, but during that conversion, you get energy lost, and that energy is usually lost as heat, and that leads to an increase in what's known as entropy. So entropy is the energy that's not able to be harnessed to perform work because it is lost as heat. So here we have an image of a car kind of showing that relationship where you have chemical energy in. So gasoline has energy stored in the bonds of those gas molecules. When the bonds are broken, that energy is used to run the car, and it turns the motor and turns the wheels, and you get the potential energy from the chemical bonds, which is gas, to be converted into kinetic energy that makes the wheels turn. This process leads to heat loss from the breaking of the molecules from the engine and the gasoline molecule, and also heat loss from the friction of the tires rolling across the ground. So again, this heat loss is adding to an increase in entropy, so an increase in kind of the chaos in the environment, because that heat dissipates into the environment, right? And that heat again comes from chemical reactions or friction between objects. And like we've noticed, molecules of matter are usually ordered, but they become disordered, right? And that constant flow of energy tends to dilute the ordering into the form of heat that again increases the amount of entropy or disorder in the environment. Some forms of energy are more useful than others. We can label them as high quality and low quality. So high quality energy can actually perform useful work, so be converted into electricity. Low quality cannot be performed to use for work. So for example, like heat in the ocean, right? So we can't really use the heat of the ocean to make energy or anything like that. And there's a temperature difference between two objects, meaning that again, heat will flow from a warmer object to the cooler object, right? And so the greater the temperature differential, the more heat is going to transfer, so the more useful work can be done. And now just because something is classified as a low quality energy doesn't mean it's not significant. So remember that heat energy we were talking about in the ocean, it actually moderates coastal climates and actually produces those cool maritime climates. And it actually leads to different weather patterns, causes ocean currents that drive fisheries, that create different climate patterns around the globe. And so again, that low quality energy still really significant for the natural ecosystems. And we also can use new ways or figure out new ways to convert this low quality energy into high quality energy. So for example, getting better turbines and different photovoltaic cells to convert low quality light and wind into electrical energy that can be used for work and high quality processes. And so in biological systems, right, we see this second law of thermodynamics. So all organisms, including us, are converting high quality energy into low quality energy. We have that high quality energy in the form of chemical bonds in our food. When we convert it into ATP and use that ATP, we make heat. So that heat is a waste that is produced, and from just a general energy standpoint, we can pretty much compare that to the process of combustion, where we burn gas, breaks the bonds, and releases heat due to that chemical reaction. The consequence of this energy conversion is pollution, unfortunately. So again, you get emissions from power plants that are converting this energy into electricity. pollutants that are released from cars. And so you, again, like we talked in the last chapter, each person has to think about their individual contribution to, um, their carbon footprint, how they're influencing the environment, and the amount of energy in the universe is limited, right? We can't create or destroy energy. It's limited. It only can be converted, and only a small portion of that energy is high quality. And so, there's this conversion cycle that that has to happen. And so once that high quality is converted, it's lost its heat, and adds to that entropy in the system. So we have to keep that in mind as we use our resources as well. Okay. And so that's it for chapter 4. As always, we have a summary here at the end, kind of let you take a look at some of the big ideas from this chapter. Again, this isn't an end all be all to what you should know from the chapter, just a good starting point for when you study for your quizzes or your exams. And so, as always, let me know if you have any questions. If you have any concerns, you can contact me in email, or you can contact me through the discussion board in the Canvas section for the course. And with that, we'll see you in the next chapter.